Okay, just a quick session on enzymes and digestion. So here are the main points of the specification. So knowledge of the human digestive system is of course important. We'll look at the structure of that. And then in particular, we're going to focus on enzymes. They're linked to the key factors that affects enzyme activity. And then we're going to discuss the specific practicals. So this is the specification. You should focus and read the key parts of it so you're aware of the key things the examiners can ask you. The practicals we'll look at, in particular, we're going to look at required practical 5, which is the effect of pH, but we'll look at some other practicals as well. So I'd say the key focus of this session really is to look at the human digestive system, to focus on the structure and function of enzymes, and then look at some enzyme practicals, and then of course finish with some past paper questions. So, in terms of human digestive system, you should be able to start from the very top and make your way down in terms of the involvement of it. In terms of digestion, so digestion is really about breaking down polymers into monomers. Polymers are of course large molecules, and the other key thing is they are insoluble. So the whole reason for digestion is to break down those polymers from large insoluble molecules into monomers. The reason we want to do that is because when they are broken down into monomers, that allows for absorption into the blood. Digestion can be broken down into two different types of digestion. We have physical digestion, which predominantly relates to muscles, and then of course we have chemical digestion, which relates mainly to enzymes. So starting at the very top, we obviously have the teeth. I'm not going to particularly talk about those. We then have our salivary gland in the mouth. The salivary gland produces salivary amylase. We're going to talk about amylase in a bit more detail later. And the whole function of salivary amylase is to start to chemically digest starch. It actually digests it into something called maltose, but then it's starting to begin the process of breaking it down into glucose. As we move down, we then get to the esophagus. Just check the spelling of esophagus as well. Esophagus plays a large role in in physical digestion, because it is a very large muscle. We then move on to looking into the stomach, which is this one. The stomach is involved in physical digestion because it has a muscle. It's also involved in chemical digestion because it produces a protease. And the protease breaks down or digests or hydrolyzes, you could say, a protein into an amino acid. It does contain hydrochloric acid. I wouldn't be talking a huge amount about the function of hydrochloric acid in terms of digestion. It's more involved in microbes and the fact it builds up a barrier to your immune system. Carrying on then, so that was our stomach we just spoke about there, so this is our stomach. As food leaves the stomach, it then enters the top part of my small intestine. So the top part of my small intestine is kind of around about here. So this is my duodenum. And where the duodenum starts, if you kind of imagine this as my small intestine, we then have lots of other parts that come into play here. So coming into play, we have a really important organ, which is my pancreas. The pancreas plays a huge role in digestion. It produces all digestive enzymes. So it produces amylase, it produces protease, it produces lipase, It does also play a role in glucoregulation because it produces the hormones insulin and glucagon, but we'll talk about that later. And also at the same point, coming in here, we have our liver. In terms of the function of the liver in digestion, is it produces bile. Bile has two functions. So the first function of bile is it neutralizes stomach acid. So if you think all of the food that leaves the stomach is at about pH 3, we want it to be at pH 7 because that provides an optimum pH for enzymes. Okay, so none of the enzymes in the small intestine would function if we didn't have bile. The second function of bile is it does something called emulsify. And in particular, it emulsifies fats or lipids. What we mean by that is this. If this is my lipid globule, it physically breaks down the lipid globules into smaller droplets. The reason for that is to increase the surface area, which means there's more surface area for lipase. And then finally, all of that food or digestive material then goes into the small intestine. And again, the small intestine produces all digestive enzymes. So if I ask you any question about where are digestive enzymes produced, the small intestine and the pancreas produce all digestive enzymes. But in addition to the digestion, it is in the small intestine where absorption also happens. Okay, so that's kind of our human digestive system, the key things to know about it. So then focusing on enzymes, just a couple of key words to talk about in terms of relation to enzymes. So first of all enzymes are proteins. And as a result of it being a protein, all enzymes are coded by your DNA or your genes. In addition, all enzymes are biological catalysts. The fact they're catalysts is two things. One, it means they are not used up. And two, it means they lower the activation energy. Which means they speed up chemical reactions. So in terms of my definition of the enzymes, that's what we are talking about. Key things then, so we talk about the lock and key theory. And what that really means is, if this is my enzyme, okay, the enzyme really is the lock in this model, and you have a particular molecule that will fit into that lock. So again, in this model, this is the key. This is my enzyme. This we call a substrate, which are basically the reactants, the things that go into it. So the enzymes and the substrate, we say, are complementary to each other, which basically means the enzyme is specific to a particular substrate. How they are specific is all due to this part of the enzyme here. So this bit okay, is called the enzyme's active site. And it has a particular 3D shape, which is complementary to a particular or a complementary substrate. So it will only fit a particular enzyme. Sorry, an enzyme will only particular substrate linked to a lock and a key. So a key will only fit in a particular lock, a substrate will only fit in a particular or a complementary enzyme. In terms of the enzymes you need to know about them, so we've already spoken about a few of them. So we have amylase. Amylase is produced in 3 places. Of course, it's produced in the small intestine, because all of them are produced in the small intestine. It's produced in the pancreas. But in addition, as mentioned earlier, it is also produced in the salivary gland. The function is to break down starch into glucose, and glucose is needed for respiration. Second enzyme, or digestive enzyme, is protease, produced in the small intestine, produced in the pancreas, but also produced in the stomach. The function is to break down proteins, which is a polymer, into an amino acid, which is a monomer. And that's so you can make any other proteins in your body, which are used for growth and repair. Here's where you could talk about particular proteins. So, you need to eat proteins so you can make enzymes, you need to make antibodies, you need to make hormones. And the third one is lipase, which is only produced in the small intestine and the pancreas. Lipase breaks down lipids, which is the proper name for fats, and that breaks it down into fatty acids and glycerol. Bile is not an enzyme, but bile very much is essential for the digestion of lipids as well. Because as we said earlier, not only does it neutralise stomach acid, but it physically breaks down or emulsifies these lipid globules into tiny little globules to increase that surface area. Factors then, there are 2 factors that affect how quickly an enzyme works. What we mean by that is it affects the rate, which effectively is often to do with the products produced in a given time. So the key thing about rate is it's over a given time. And there are 2 factors. So one is temperature, the other one is pH. So if we look at temperature, you need to be able to describe and explain the effect of temperature on the rate of enzyme reactions. So just listen to how I describe it. As temperature increases, the rate of enzyme activity equally increases up to a certain point, and then it rapidly decreases. So key things here. This is called the optimum, which is the best temperature. So as I said there, as temperature increases, the rate increases slowly up to a certain point, then it rapidly decreases. Okay, you need to be able to explain why. Well, as temperature increases, the amount of kinetic energy increases, which increases the number of collisions between the enzyme and the substrate. So as they have more kinetic energy, the likelihood of those enzymes and substrates colliding together increases, which means the rate of reaction increases. At a certain point then, so beyond the optimum temperature, the enzyme becomes denatured. What we mean by that is this. If this is my enzyme, it then starts to look like that. So what does denatured mean? It means the active site changes shape, which means the enzyme is no longer complementary to the substrate. Then you have pH. So different enzymes work at a different pH. So obviously again we have pH on my x-axis, we have rate on my y-axis. The protease that works in the stomach has an optimum pH for about pH 2. But most other enzymes have an optimum pH of about pH 7. You can see that for pH, you have an optimum pH, but either side of the optimum pH is symmetrical, and either side of the optimum pH the rate decreases, and that's due to either side increasingly becomes denatured. There's the main practical you need to be aware of is the effect of pH on the enzyme amylase. Okay, again, if I ask you to write a method, always start by just listing the independent variable, the dependent variable, and the control variables, ideally 3. So the independent variable is the pH, because that is what you're changing. The dependent variable is the rate of amylase, but I would also include what I'm going to talk about in a second, which is actually what you're recording. The control variables are, of course, temperature, but I would also then include the concentration of amylase, and I would say the concentration of starch. So what you're going to have is this. You're going to have a solution, and in that solution you're going to have starch, you're going to have an exact volume, and you're going to have an exact concentration. You're then going to have amylase. You're going to have an exact volume, and you're going to have an exact concentration, because they're control variables. And to control the temperature, this is all going to be carried out in a water bath. So you let them reach the correct temperature, so they're both acclimatised to the correct temperature. And then what you would do for this practical is you would set up a white tile, and in that white tile you would have a couple of drops, again 2 to 3 drops to keep it valid, of iodine. And every 30 seconds you would take a drop out, so this would be 30 seconds, this would be 60, 90, 120, 150, 180. And you're looking to see the time taken for the solution to no longer go black. If it goes black, so black means that starch is still present. If it does not go black, it means that starch has been broken down into glucose. The other practical just to be aware of is they could ask you to do obviously the effective temperature. And all you would do there is just have a range of temperatures in a water bath. And of course you need to keep all the other variables, in particular pH, the same. The hardest one they could talk to you about is looking at lipase. Because lipase breaks down lipids into fatty acids, and glycerol, of course, they could be asking you how could you record the rate of reaction for the breakdown of, it could be the effect of pH, it could be the effect of temperature. Because it's fatty acids, what you're going to see happen is the more lipids that are broken down, the pH is going to go from 7 to, say, 5. And it'll allow you to monitor the pH. And the reason the pH changes is because as lipids are broken down, you're producing fatty acids, which is making the solution more acidic. So you could use a pH meter, or you could use an indicator, in particular something like phenolphthalein, which will change colour when the pH has changed. So just be aware of particular questions they could ask you about that. As per usual then, a couple of past paper questions. So what you should do is you should go to the past paper question, pause it, attempt the questions. Once you've attempted the question, attempt the next question. Take a look at the mark scheme, 5 marks for this one. And then check the mark scheme, see if you've got all the valid points. So this is A, B, C, and D. And then E, F, G, H there. Pause it. This is a really good question actually here, talking about effect of pancreas. So why do they lose weight? So do not refer to hormones in that question, so pause it. Here's then the mark scheme for this one, so a really good one here about enzyme production. I'd have a go at that one. Bit of data analysis question. So student investigate the effect of changing pH and temperature. So this one is changing pH, this one is changing temperature. So you'll need to use that. I've included the same data here. 2 questions. Lactose is normally produced in the stomach, and how do you know that? We'll look at the pH. Explain as full as you can the results in table 2. So that's the effect of temperature. And this is then the mark scheme for that. 2-mark question on bio. And that should be it. Thank you.